Okay, good afternoon, everyone. Welcome to the College of Pharmacy seminar. I'm standing in the stead of Dr. Kumar for today in terms of chairing this seminar. It is my pleasure to have Dr. Botchwey, one of my former professors from Georgia Tech, speaking at the seminar. So just a brief introduction about Dr. Botchwey. Dr. Botchwey is a professor in the Wallace H. Coulter Department of Biomedical Engineering at Georgia Tech and Emory University. His research focuses on advancing regenerative medicine and immunoengineering through a convergence approach that integrates various disciplines and techniques for tissue repair and regeneration. Dr. Botchwey's lab has made significant contributions in investigating S1P signaling in tissue repair and engineering in vivo gradients for microvascular remodeling, as well as developing regenerative immunotherapies using specialized pro-resolving lipid mediators, SPMs, and hydrogel-based delivery systems. This talk will mainly focus on targeting S1P signaling to promote tissue repair and regeneration, including the use of sphingosine kinase inhibitors to reduce local S1P levels and promote muscle fiber regeneration. Additionally, the development and application of APO-M, FC-S1P, a novel S1P chaperone that enhances S1P receptor 1 signaling will be discussed, highlighting its role in reducing fibrosis and improving muscle healing, particularly in aged animals. Moving right along, Dr. Botchwey aims to develop innovative, equity-centered solutions to complex challenges in regenerative medicine with potential applications in diverse fields such as regenerative immunotherapies, metabolic and hematologic disorders, and cell and tissue transplantation. Additionally, he is actively involved in promoting diversity and outreach programs like the SURE program, which I was a beneficiary of, and engaging new generations at Georgia Tech through engineering and science, which aim to provide underrepresented students with research opportunities and inspire the next generation of scientists and engineers. Please welcome Dr. Botchwey.
Thank you so much, David. It's really a delight to be able to participate in this way. And thank you also for your accommodation for me to do it virtually. Um, I haven't visited the campus before, even though I went to University of Maryland, College Park. And I
Okay.
did want to, at some point, be able to reconnect with you in person, but, perhaps another time, and let's make the most of, this opportunity I have with the students. So I just appreciate everyone's time today. So I wanted to talk a little bit about a piece of my work. I'm trying to tailor it to an audience that, in kind of the pharmacology realm. My background is in biomaterials and regenerative medicine. And, if at any point I'm, maybe, going off or explaining things that, in a context that is maybe difficult to understand, I have no problem taking questions along the way. But
Dr. Botchwey, we can't see your slides.
you cannot see my slides?
No, cannot see your slides. Nope.
Okay. Let's see.
Okay.
How about
There
now?
we go. Yeah, we good now.
Okay. Do you see the next slide?
Yes, sir.
All right. Very good. So, like I said, I'm a biomedical engineer, so I'm interested in the body's capacity to regenerate. And, historically, we can compare species like salamanders and human beings to see just how divergent the underlying biology can be in terms of how we heal and what capacity we have to regenerate after significant injury. So, um, many of you may be familiar with the idea that salamanders can regenerate a whole limb perfectly. Skin, bone, muscle, all the different compartments of tissue. Whereas in human beings, we scar. And just historically, if you look at just some of the milestones where we've begun to understand some of these key differences, we can kind of see early on cells are activated in a way that starts a cascade of signaling, where ultimately the epidermal cells, fibrotic cells that make extracellular matrix, nerve cells, cells that make new blood vessels, act in coordination to reform limbs, whereas the stage is set fairly early on after traumatic injury that humans are going to start to form a scar, right? And so the end result of the salamander is complete limb regeneration. For us, it's scar. And we're interested in models where these types of divergent outcomes bear themselves out. So I'm going to be talking today primarily about a model of volumetric muscle loss. So this is a significant traumatic injury where there's a frank loss of muscle tissue. And just to give you kind of an example of how this plays out in actual clinical applications, here's just a severe case of traumatic injury where and I spared you just some of the gory images of the soft tissue damage in favor of just some of these radiological images where you can appreciate how much trauma this particular limb has sustained and how much surgical intervention is required to reconstruct this limb and try to salvage it. But what I've listed here are just some of the classic outcomes that you see after traumatic injury. So donor site morbidity refers to the fact that oftentimes you have to transplant muscle from one place to another in order to fill gaps to get this tissue to reconstruct this limb and to salvage it. Oftentimes, that tissue is subject to fatty infiltration and fibrosis, which ultimately limits the functional recovery in the injury. And significant scarring is also going to lead to kind of a physical deformity. And so this is a fairly significant problem. And it's really akin to the challenges of reforming a limb because there are multiple cell types and processes in the healing cascade and tissue types involved. And so we've tried to study this kind of injury at a preclinical level so that we could come up with interventions that can address some of the root causes and ideally unlock regenerative potential that may be there, but is not properly harnessed in humans. And so this is a paper that we published of our mouse model finding. So essentially, this is a picture of the mouse quadriceps muscle, and if you excise about 20% of that muscle, which ends up being close to about a 3 millimeter punch biopsy injury into the quadriceps, you replicate many of the hallmark features of the human clinical VML injury. And this is just giving you one indication that that the strength recovery after this VML injury is very similar to the strength loss that you see in humans. Similarly, you don't get the bridging of myofibers across this gap that you can see kind of outlined in this cellular region in the 3 millimeter defect. As well, you see some of these streaks of fibrotic tissue that are infiltrating the injury microenvironment. And you also see these CD68 positive immune cells that persist in the injury long after inflammation should be resolved. And this was really one of the keys for us. That is to say, to what extent might the failure to resolve inflammation be one of the critical determinants of what kinds of injuries will scar in comparison to what kinds of injuries will heal? And if you actually take a look histologically in our model system, that again, we can, in this case, look at divergent outcomes of critical injury that replicates the human VML outcomes versus what we call subcritical injury that mimics the type of injury only at a smaller volume. By subcritical, we're implying that that muscle will heal and inflammation will be resolved. And we won't get the fibrotic scarring that we get in the critical defect. And so in our model system, we can make comparisons about what is different about the critical size injury and what can we learn from it that could form the basis of an intervention that targets some of this persistent inflammatory mechanism, right? And so essentially what you're seeing is that there's a certain subtype of macrophage called M2 macrophages. This is a polarization state that can contribute to things like angiogenesis and inflammation resolution. But if these macrophage subtypes persist in the injury too long, they can actually become activated to states that are signaling toward more fibrosis and scarring. And indeed, you see an increase in a persistence or an increase in the number of macrophages in the critical size defect 7 days after injury, where you really should start to see some of those macrophages egress and or be removed from the defect site. And if you use second harmonic imaging to look at what you see in blue are the collagen microfibers that are being laid down by fibrotic cells in each of these scenarios. So this is an uninjured muscle where SHG is just showing us the collagen fiber signal. Red is showing us those M2-like macrophages. And green are fibroadipogenic progenitor cells that we refer to as FAPs. And essentially, I'm condensing a lot of work here that we published last year in a paper in Communications Biology. But essentially, this is forming an interaction of or an intercellular communication where these profibrotic macrophages are signaling to these fibroadipogenic cells or FAPs in ways that starts to lay down this disorganized scar-like collagen network. And so this is essentially laying the groundwork for more fibrotic outcomes that we associate with VML injury. And indeed, you can quantify that you see many more of these types of crosstalk circuits in the critical size defect, and others have published. So this over here is not our paper, but I just wanted to give you a sense of scale. What you see in blue are the mononuclear phagocytes. Those are the monocytes and macrophage cells that are persistent in injury. And if you look in the defect area, so this is another laboratory, but I thought that this is very illustrative. By day 7, over 90% of the cells that are in the injury that we created in this muscle are monocytes and macrophages. And so that got us thinking of ways in which we could target those macrophages so that we could perhaps work on how to clear them from the injury site and or induce them to adopt states that are actually signaling in ways that promote healing as opposed to scarring. So we've done some single cell RNA-seq analysis. What you're seeing here on the left-hand side of my screen is what we refer to as a UMAP. So this is a dimensionality reduction technique that allows us essentially to map where the clusters of cell types are in the injury microenvironment at that critical day 7 time point. And we can isolate those macrophages. And because we included both the critical size defect data as well as the subcritical size defect data, we can make quantitative single cell comparisons of what the gene expression level differences are between macrophages in the subcritical injury compared to the critical. And as I'll simplify the explanation of what you're seeing is that if I isolate macrophages and I use gene ontology analysis to tell me what kinds of genes tend to be expressed in macrophages that can be isolated or derived from the subcritical injury, what we see is that these macrophages express high levels of genes that are associated with migration and lower levels of genes that are associated with adhesion. The converse is true of critical size defect. And the result is that in the critical size defect case, macrophages are expressing genes associated with migration at a lower level and are expressing genes associated with firm adhesion at a higher level. And for that reason, their egress from the defect site in comparison to those macrophages in the subcritical injury is much less robust. And so that's one mechanism that can contribute to their persistence. And if you do flow cytometry analysis and you isolate those macrophages from the defect at that same day 7 time point, you can also see that this is confirmed in what the cells actually are generating and secreting. So this is an intracellular cytokine staining for both TNF alpha and TGF beta, which are known signals that are driving kind of fibrotic signaling and crosstalk with those fibroadipogenic progenitor cells. And so the macrophages are expressing pro-fibrotic signals at a much higher level in the critical size defect. So this begins to form the basis for our thinking of regenerative immunotherapies that can target these macrophages and either aid their egress from the defect site so that the processes of healing can proceed without that excess profibrotic signal, or can we alter the states of those macrophages so that they begin to signal in ways that are promoting healing? Or can we achieve both of those things? One of the specific targets that we had in mind because of expertise we had in our lab was targeting the sphingosine 1-phosphate receptor signaling pathway. So S1P is a bioactive lipid, is a pleiotropic lipid mediator that is derived from the metabolism of plasma membrane lipids like sphingomyelins and ceramides. And much like you think of growth factors and cytokines, these lipids, these bioactive lipids, have cell receptor, have plasma membrane receptors. So S1P receptor 1 through 5 are the 5 high affinity G-couple receptors that recognize this native S1P ligand. And it is involved in a wide variety of cellular functions. And so I'm just going to quickly orient you to some of them. So S1P is intimately involved in the trafficking of immune cells, both lymphocytes and mononuclear phagocytes. And so at homeostasis, tissue levels of S1P are actually very low, but in and during inflammation, you actually see the generation of S1P such that gradients of S1P are actually driving the influx of immune cells from the blood into different tissue compartments. And so S1P can regulate immune cell trafficking. S1P is probably best known for its role in regulating barrier permeability in endothelial cells. And so this is just an illustration of how tight junctions between the epithelial cells in the blood vessels is very important in kind of maintaining the barrier integrity. And so S1P is one of those molecules that can open and close those junctions between cells. And interestingly enough, S1P can actually play a role in both increasing inflammation by opening those barriers up when it's chaperoned by albumin molecules in the blood. And it can also play a role in helping to restore that barrier when the same molecule is chaperoned by the ApoM domain of this HDL microparticle in the blood. So I'm going to come back to this later on in my talk, but I just want to give you some orientation to the fact that S1P is chaperoned in the blood, and it actually matters what molecules, what proteins in the blood chaperone this interaction with S1P receptors on endothelium. And then lastly, S1P can play a direct role in signaling to profibrotic cells. I'll keep it straightforward. When you have damage to the blood vessels, as well as some tissue structures, S1P can play a role through several of its receptors in promoting extracellular matrix deposition from the fibrotic cells and inducing some of these immune cells into profibrotic states. And so S1P has a role in a number of the processes that we are concerned about in VML. So now, can we begin to think about S1P and its regulation of these important processes in regeneration as a potential target, as a strategy for harnessing the functions of this molecule in a way that can promote healing, right? So that's what the rest of my talk is going to be about. So this is a kind of a complex picture of this metabolic process that I talked about. All it's illustrating is the fact that here's our plasma membrane. This is the S1P receptor. I mentioned that plasma membrane components like sphingomyelin can be metabolized by various enzymes that can convert sphingomyelin to ceramide, ceramide to sphingosine, and sphingosine kinase converts sphingosine to S1P. It's actually transported back across the plasma membrane for an autocrine-like signaling to S1P receptors. That's why this is drawn in kind of a loop. But what I want to draw attention to is the fact that we can use small molecule therapies to target some of the enzymes responsible for the production of S1P at any of these stages. But I want to zero in on some molecules that are available, at least for research purposes, that can inhibit the activity of sphingosine kinases. So the conversion of sphingosine to the bioactive form of sphingosine 1-phosphate. So let's just talk about ways in which we can kind of target the generation and metabolism of S1P as a form of regenerative immunotherapy. And I'll make this one quick because there are a couple of molecules that that we have available to us. But before we even get there, there's also an antibody that can just confirm something for us, which is to say S1P does persist in its concentration in the defect site in the critical size injury. And so basically what you're seeing are histological sections where we've just stained for the molecule at this critical day 7 time point. And this is just a visual representation of the fact that S1P generation is significantly greater in the critical size injury condition. So that's further justifying S1P as a potential target in VML specifically, apart from all the things we know about how this molecule functions. So here again, we're targeting sphingosine kinase, and there are two isoforms of the enzymes, sphingosine kinase 1 and 2. They're actually small molecule inhibitors of both of these enzymes. And so we just did a relatively straightforward experiment where we did intraperitoneal injection of the small molecule in our mouse defect VML model and we did kind of repeated injections of the molecule over about a 14-day time course. And I'm just going to show you just some of the histological data that we see. So what we can confirm is that in the case where we're just injecting our vehicle control, we see large amounts of positively stained S1P in the defect region that we created in the muscle. No surprise there. We had seen that before. These are the two cases in which we did IP injection of the sphingosine kinase 1 and sphingosine kinase 2 inhibitors. And we can see that there appears to be a reduction in the amount of S1P in the muscle. However, there's some interesting, kind of pharmacokinetics that happen with sphingosine kinase inhibitors that I probably don't have enough time to explain in great detail. But for those of you who may have been following along from the slide and getting ahead, basically what this is showing is that when you inject a sphingosine kinase 1 inhibitor, as you would expect, plasma concentrations of S1P actually go down. Conversely, if you inhibit sphingosine kinase 2, plasma concentrations of S1P actually go up, which is really interesting because sphingosine kinases, in both cases, are producing or converting sphingosine to S1P. But regardless, what was important for us is that S1P levels in the tissue do go down in both cases. This one kind of trends down with sphingosine kinase 2, but we see a significant reduction in S1P concentration in the muscle after sphingosine kinase 1 delivery, much like you would expect. And if we look for the histological evidences of how the muscle is healing over the course of 14 days after we deliver these sphingosine kinase inhibitors, you can see that here muscle fibers are outlined in this with this dystrophin antibody. We stain for embryonic myosin heavy chain as a marker of the early formation of new muscle fibers. And DAPI is going to stain for the nuclei of cells that can give us a picture of the cellularity in the defect site that also is often an indication of persistent inflammation, right? So this is an image of what the untreated muscle looks like. David, did you want to interject with something?
No, sir. No, sir.
Okay. All right. And what you can see in the sphingosine kinase 1 inhibition group is a significant increase in the new muscle fiber formation, which is trending in a direction much more toward regeneration than scarring in comparison to the untreated PBS control. In sphingosine kinase 2, you see what could be characterized as a somewhat more a better healing outcome in comparison to untreated, but it's a pretty clear distinction that many more new muscle fibers are actually being formed in the sphingosine kinase 1 treatment condition. And this is if we go back to our RNA-seq data, what we can see is that when you look at the average expression level of sphingosine kinases, both isoforms, in cells in the defect area, what you can see is that there's a much higher expression of the average expression, as well as the percentage of cells that express sphingosine kinase 1 in comparison to sphingosine kinase 2. So what this is essentially aligning with is the idea that sphingosine kinase 1 inhibition has a much more significant effect at reducing S1P concentration in the defect site. And this may be attributed to the fact that the expression level of in the untreated condition of sphingosine kinase 1 by cells in the defect is much higher. And so in some senses, sphingosine kinase 1 inhibitors are targeting the root cause of this metabolic dysregulation of S1P generation in comparison to sphingosine kinase 2. And that actually is consistent with the healing outcome that we obtain. So that was kind of a quick story. And so I want to switch gears to another strategy in which we could take advantage of the selective agonist and antagonist for S1P receptors that are available. So there are some approved drugs now. So if you're familiar with Gilenya, which was the first in class S1P receptor targeted immunotherapy for relapsing remitting multiple sclerosis that was approved several years ago. Its investigational name was FTY-720. And the success of that molecule actually spawned kind of drug discovery programs in all the major pharma companies looking at various agonists and antagonists of S1P receptors, because FTY-720 is an agonist of S1P receptors 1, 3, 4, and 5. It has no activity at S1P receptor 2, but more importantly, when FTY-720 signals to S1P1, you actually get an internalization of the receptor ligand complex. The receptor is ubiquitinated, and it's called what's called a functional antagonist of S1P1, because the receptor is no longer recycled back to the plasma membrane. So you can essentially remove S1P1 availability on immune cells when you deliver FTY-720. And many different pharma companies were trying to exploit this phenomenon by developing S1P receptor targeted molecules. If we go back to our RNA-seq data, we see that in the clusters of endothelial cells in this case, that are mediating kind of immune cell recruitment, we see different patterns of S1P receptor expression. And interestingly enough, that S1P receptor 1 that can be functionally antagonized by S1P receptor targeting molecules, you actually see a high level of S1P receptor 1 expression in the critical size defect endothelial cells. So that also gave us some hope that we could actually exploit approved immunotherapies in the context of our VML model. However, we got actually interested in S1P receptor 3 for a variety of reasons. Maybe the quickest summary is if you do make a defect in S1P receptor 3 knockout animals, we actually see, this is back to our embryonic myosin heavy chain expression level, you actually see a pro-healing phenotype where many more new muscle fibers are actually being formed in the S1P receptor 3 knockout mouse, right? So that gave us some inkling that if we had small molecules that could inhibit S1P receptor 3, we might be able to replicate some aspects of this muscle healing promotion that we saw in the knockout mouse. We knew of a molecule that would inhibit S1P receptor 3. That molecule is called VPC-01091. And so similar to what we did with the sphingosine kinase 1 antagonists, sphingosine kinase antagonists, we did an IP injection of this VPC-01091 molecule. And we had published previously that it had activity in ways that mobilized stem cells from the bone marrow. And this is just some of the data that we showed about the fact that it can target S1P receptor 3 expressing cells. And in the case of both hematopoietic stem cells and mesenchymal stem cells, inhibiting S1P receptor 3 actually loosens them from their niche in the bone marrow and allows them to mobilize into peripheral blood. But in this case, we're using it as a potential target of cells in VML injury. So we do a couple of injections at the time of surgery and about 6 days after the surgery. And then we take the experiment out to 14 days and we isolate both tissue and blood and do our analyses. And this was a very, very potently effective treatment. So here again, what we're seeing is our molecule, the untreated condition where you see, kind of this unchecked significant level of inflammation that you see at day 14, and a significant scar that is forming in the untreated muscle. And in the case of the VPC-01091 injection, we actually see a fairly potent pro-regenerative type of outcome. And this is borne out in the numbers of new muscle fibers that are being formed by EMHC staining. Also, we can also quantify the growth of those newly formed muscle fibers by this metric that we call centrally located nuclei. And so essentially we're just counting the number of these muscle fibers that have these nuclei in the center and the size of those new muscle fibers. And basically what we're seeing is that the area of centrally located nuclei, or the area of more mature muscle fibers, is also greater. So more muscle fibers are growing, and the growth of those muscle fibers is far greater in the case where we conditioned with this VPC-01091 based immunotherapy. Another couple of examples is we can also create biomaterial strategies where we can.
Did you get anything printed?
Is there? Okay. Was that a question? Maybe not. Okay. So we can also create biomaterials where we can locally administer these drugs so that we don't have to do multiple injections. And in this case, we used a polyethylene glycol base. This is a 4-arm PEG monomer that is terminated with norbornene groups such that we can use a photoinitiator and these thiolated crosslinks and take advantage of click chemistries where we can put in this PEO is just kind of a water-soluble space filler for the fabrication of the scaffolding. We can put adhesive ligands in the molecule as long as it's thiolated to react with these norbornene groups. This is our PEG dithiol crosslinker, so that by mixing these things together with our photoinitiator and applying light, we can actually react these monomers together in a way that forms a hydrogel, right? And that hydrogel can actually use these different crosslinks as well as the adhesive ligands to create essentially a form of matrix or synthetic matrix that cells can interact with. They can attach to these adhesive ligand signatures. We can deliver drugs from this network and take advantage of any interaction that the drug might have with molecules that we embed in the polymer backbone. So this is a very robust technique of scaffold fabrication that has been used in a variety of different applications by the Garcia lab at Georgia Tech. Andres Garcia is a colleague of mine at Georgia Tech and in IBB, one of our primary interdisciplinary institutes. So in this particular example, I just want to show you a case in which we took this scaffolding approach at hydrogel fabrication, and we simply just encapsulated the drug inside of this polymer network and allow it to release via diffusion. And so we can implant the gel into the defect and see what the healing outcome is. Now, there's one other layer that I want to introduce to you, which is we can not only can we make bulk hydrogels that you can kind of visualize in this gumdrop looking illustration, but we can also fabricate those hydrogels in the form of these extracellular matrix-like or extracellular mimetic fibers. So this is a process called electrospinning. So we just take this polymer solution, we apply a voltage drop, and we collect it in a grounded collector plate. And because of the electric field forces that pull the polymer solution or the droplet apart as it's kind of whipping around in this working distance, what you collect is a meshwork of fibers that we can essentially apply light to fix this fiber-like network in place. And the organization of these fibers is somewhat like extracellular matrix, right? And so we can actually add a layer of organization to the scaffolds that we create by building in these type of geometries that mimic the extracellular matrix that many of these tissues are composed of. And we can also use it to tailor the release profile of the drug at the same time. So I'll just show you some data where we just use this strategy of electrospun fibers that are encapsulating the VPC-01091 drug. We did our surgery, we implanted the defects, and basically what we actually compared bulk hydrogels, which have no fiber geometry, to NF is referring to the nanofibers. So we're referring to the electrospun mesh as a nanofiber network that's releasing our drug. And, it's one of the things I wanted to point out, even before we get to the drug-treated outcome, is that here is just a histological section of the outcome that you get when we implant just the bulk hydrogel alone, right? So you can kind of see that this healing outcome is not great, right? So just the vehicle alone was not as effective as a therapy. However, the nanofiber geometry of the same basic drug delivery vehicle tended to promote some level of healing even before we released the drug. And what we were excited about is, remember, part of what we think is the root cause of VML is the persistence of some of these M2-like macrophages in the defect. And if you actually compare the number of M2-like macrophages, so this is not in the tissue, so these are the number of M2 macrophages that have egressed from the tissue into the draining lymph node, right? So we took the lymph node, we did our flow cytometry analysis, and essentially what this is showing is that the migration and egress of macrophages from the defect area in cases where we implanted our electrospun fiber scaffold is significantly increased. And so we can mitigate some of that persistence simply by implanting the scaffold. Similarly, if we look in the tissue, we can see a persistence of these cells in the bulk hydrogel group in the tissue, whereas we can reduce the number of macrophages that are in the tissue simply by introducing the nanofibers. And when we treat with the VPC-01091 drug, we can capture some level of synergistic increase in the number of centrally located nuclei and other metrics of muscle defect healing. So the combination of the scaffold geometry alongside the local delivery of this immunotherapy tends to significantly increase the formation of new muscle fibers and their growth in the defect after VML injury. So I'll show you maybe one or two more examples before I close. And so I had mentioned previously that S1P itself is actually chaperoned by proteins in the blood. And I mentioned that it can be chaperoned by albumin protein in the blood, which is abundant in plasma. But it can also be chaperoned by the ApoM lipoprotein on the HDL particles in the blood. Now, one strategy that we've employed in collaborations with the Tim Law's group at Boston Children's Hospital in Harvard is that he made a fusion peptide where he fused the FC region of an antibody to the ApoM domain of this to the ApoM lipoprotein. And so essentially, instead of the HDL particle presentation, he's just increasing the ApoM chaperone bioavailability using a long-acting and long circulating form of this fusion peptide. And so in that way, you can take advantage of something. Basically, what this is showing you here is that after the endothelium is damaged, albumin presentation of S1P is actually promoting the activation of endothelial cells and the opening of the endothelial barrier, which is one of the critical stages of inflammation. And as I mentioned in my opening, the ApoM presentation of S1P actually does the opposite. And in some models of both kidney and lung fibrosis, Tim Law's group had shown that the administration of this fusion peptide was actually anti-fibrotic and prevented kidney injury-induced fibrosis and lung injury-induced fibrosis in some preclinical models. And so because fibrosis is also our enemy, in the context of VML, we reached out to them and they were gracious to collaborate with us to test how this chaperone protein could actually be utilized in the context of VML immunotherapies. And I'll skip over this. Just the upshot is that the basis for the development of this ApoM FC fusion peptide was that both in lung and in kidney, S1P1 receptor expression level is significantly reduced. And they wanted to be able to target lower level expressing S1P1 with a chaperone protein that would, have a higher affinity for what S1P receptor 1 could restore barrier integrity. The same kind of pattern of S1P receptor 1 expression exists in VML. I'm not going to get into that to any great detail, but we did our VML injury. We administered the fusion peptide a couple of times over the course of a few days, and we were very encouraged by the outcome. So what you're seeing here is the ApoMFC TM is a fusion peptide, but the binding domain of S1P has been mutated in a way that the molecule is physicochemically similar to the ApoMFC peptide, but it no longer binds S1P. So the TM refers to, it's a triple mutant peptide that doesn't bind S1P. And so it doesn't have any kind of therapeutic effect in the context of VML. However, we got a significant reduction in fibrosis of the muscle when we administered the active ApoM-FC peptide. In other words, providing the increased availability of S1P signaling through the ApoM chaperone protein using the fusion peptide significantly reduced fibrosis, and also increased the numbers of new muscle fibers and reduced the irregular kind of muscle fiber organization that we see in VML. Another way to look at it is with this Mason's trichrome staining as well. And so we see both the cellularity in the defect space as well as just the number of new muscle fibers is significantly reduced by the ApoMFC peptide. So we're encouraged by this type of approach. And another kind of notable thing about VML that is probably less it may be you may be less interested right now, because I suspect that many of you on the call are pretty young, but as you get older, you start to care about things like how much regenerative capacity we lose as we get older. Muscle in particular loses regenerative capacity the older we get. And so one of the things that we wanted to look at is whether or not any of our regenerative immunotherapies would actually be effective in older animals, so that perhaps we can, tailor strategies that can be effective for older, more mature, or even elderly populations of patients that suffer these muscle injuries. And so we took a cohort of aged animals that were, for a mouse, that is, that can range between, let's say, an elderly mouse is between 18 months to 24 months old. That's pretty old for a mouse. And we did our same VML defect injury and we wanted to see what the outcome was. Now, for this, we actually wanted to do some longitudinal type of assessment of changes in the properties of the muscle and any fibrosis during the course of the healing. And so for that, we collaborated with the Emory lab at Georgia Tech. He's an expert in ultrasound-based technologies. And so the technique that I'm showing you here is called ultrasound elastography. And this is supposed to be a video that's not playing. I won't worry about it. But essentially, he's applying a focused ultrasound pulse into the muscle. And what you're actually looking at in this color-coded image is the shear wave that propagates through the muscle. And you can actually measure some of the properties and the velocity of this shear wave propagation through the muscle and convert it to mechanical properties of the tissue, which is important. And so it's a way in which we can actually measure the stiffness of the tissue over time. And so we were pretty excited about some of the outcomes of this study insofar as, in comparison, here we have PBS control, triple mutant, S1PFC, and the long and the short of it is that stiffening of the muscle, which is kind of indicated here. I'll just kind of condense a lot of the data that we have. This is untreated control. The muscles get very stiff. And if you treat them with the ApoMFC peptide, you actually get a significant reduction in muscle stiffness. And you also see a significant reduction in the amount of fibrosis that forms in aged animals. So all this is to say that the fibrosis reduction is as effective or more in aged animals as it was in young mice. And so we were excited about the potential of this particular immunotherapy, even in the context of healthy aging and recovery from trauma. I had one other story about how we can do regenerative immunotherapy in another problem called oral nasal fistula reconstruction. So many of you might be familiar with cleft palate as a clinical problem. So this occurs in about 1 in 1,000 live births. So this little guy has to undergo quite a few numbers of surgeries in his young life. And this exterior reconstruction can leave a small residual scar after the surgery. What you often find is as these kids continue to grow, you get a recurrence of this fistula, right? So this connectivity between the oral cavity and the nasal cavity that has to be essentially reconstructed again using things like allografts, implants to kind of cover that fistula. And so the problem of oral nasal fistula recurrence after this cleft palate repair surgeries is a very significant one. So maybe I'll condense my story into maybe 2 slides or 3 slides, which is we wanted to know whether we could take one of the currently approved S1P receptor targeting drugs in order to achieve a guided tissue regeneration after ONF reconstruction surgery that would mitigate the recurrence of the fistula. So we did a couple of things. We used a different polymer, but the same electrospinning strategy to embed FTY720 into a nanofiber matrix. FTY720 we chose because it is a currently approved drug. The one that I referred to in the talk, the VPC-01091 drug, is not yet approved in human trials. And so we wanted to collect the drug. And this is just showing you, this is just atomic force microscopy testing, just showing you that the morphology of the fibers, as well as their physical properties, aren't changed in any significant way by encapsulation with the drug. This is some data that we had published previously about how locally administered FTY720 from nanofibers can actually change the composition and function of mononuclear phagocytes. And we had published this over 10 years ago in some cutaneous models of injury, but we hadn't tried it in this oral nasal fistula model. So we developed a model system again in mice where we actually used thermal ablation to create a fistula in the mouse. It doesn't heal. unless we intervene. And I just wanted to show you quickly some data where we embed a nanofiber scaffold that's releasing this S1P receptor targeting drug called FTY720, or we just deliver the unloaded scaffold control, right? We put a tegaderm matrix or a glue just to hold the nanofibers in place after the surgery. And so we were encouraged to see that just histologically, the FTY720 group, what you can see in the dark red here in these stains is that what you want to see is a layer of bone that gets formed and a re-epithelialization of the tissue. And you can see that actually occurring very, very well, a restoration of the normal structure of the palate in these mice when we treat it with the FTY720 releasing matrix. And I'll let this be my last slide. It's a little easier to appreciate here. So this is just showing you that essentially what the defect area looks like with and without treatment. And so you can see that after 7 days after the creation of the injury, you can still see the fistula in the untreated group. You can still see the fistula in the group where we implanted the scaffold with no drug. And you get almost a complete restoration of structure and function in the group where we are delivering even transiently this biomaterial scaffold releasing FTY720. And that's actually borne out if you measure the defect area. So we're excited about that result and its translational potential, possibly. So just in conclusion, I primarily focused on VML, significant challenges for both civilian and military populations with this injury. We hope that some of our regenerative immunotherapies can make an impact in this important clinical problem. I showed you several kind of ways in which S1P-based signaling can be exploited, from inhibiting its production to kind of targeting the proteins that chaperone it to bias toward more inflammation resolving signaling. I showed you the effect of nanofiber scaffolds in delivering the drug and how that can be so beneficial in injuries where the trafficking of immune cells in and out of the defect is important. And I even showed you that these kinds of approaches that were borne out in VML can also translate to other types of injury microenvironments. In this particular one, I showed you kind of this cleft palate repair, or the problem of oral nasal fistula recurrence after cleft palate repair surgeries. And so we're excited about the future here. And, I just want to, point out the outstanding graduate student who's, I should update my slides. She's now a Ph.D. that's working in a postdoc in Belgium, Lauren Email, who did the majority of the work that I showed you. The work that I showed you on oral nasal fistula was done in collaboration with my colleague at Children's Healthcare of Atlanta, or CHOA, and Emory University, Dr. Stephen Gowdy and his student Toma Afra, and of course my funding sources. And so I, thank you again for the opportunity to present to you guys. And if there are any questions, I'm happy to field them.
Thank you again, Dr. Botchwey. I highly appreciate this. Talk was very interesting. I'll open the floor for questions, but I have a few if there are none. Okay, so hearing none, feel free to unmute if you have a question. Okay, cool. So hearing none, Dr. Botchwey, what implications do you think your work has for potentially neurodegenerative diseases? I know we're dealing with muscular regeneration, but this technique or this strategy of regenerative medicine, what implications do you think your work may have for neurodegenerative diseases? Even though the work that you discussed seems very important in terms of its regenerative capacity, especially when it comes to cleft palate and these other types of signaling pathways. What implications do you think, or what insights do people glean for the treatment of neurodegenerative diseases?
It's a great question. The most straightforward answer for the treatments that I talked about today is that I don't know yet. However, I'll share with you an anecdote. So there's another part of my laboratory that does research in sickle cell disease. Mm-hmm. And one of the kind of underappreciated complications of sickle cell disease is learning and cognitive defects that can manifest themselves in, in fairly young ages. And so I had a colleague who's at Cincinnati Children's Hospital reach out to us to do lipidomic profiling, similar to what I showed you with the antibody labeling in VML, just to see if some of the bioactive lipids are dysregulated in the brain in sickle cell disease. And indeed, hopefully, some of our work is about to come out for publication. But indeed, some of these same bioactive lipid pathways in the context of the learning challenges in kind of preclinical models of sickle cell disease suggest that some of the same kind of immune dysregulation rooted in bioactive lipid dysregulations also plays out in the brain in various contexts. And that got us thinking about the broader implications for degenerative diseases as well. And so we may end up moving in that direction of neurodegeneration, but we'll probably only get at it through the lens of sickle cell disease treatments. But we're excited to see where that goes.
Okay, Dr. Botchwey. Well, it seems like we have near the time. So thank you again, and I highly appreciate you taking the time to give this talk on behalf of Dr. Kumar and all of the professors on the Research and Professional Development Committee. We appreciate your time, and I hope to hear from you soon. Any further questions? You can reach out to me by email, and I'll connect you with Dr. Botchwey. Thank you again, Dr. Botchwey.
Okay, thanks everybody.